Okay, if at any point I start mumbling, please raise your hand and say it. And it goes without saying that this is meant to be instructive, so you can stop me at any point, ask any questions that you would need to ask, because that's what's expected in this sort of setting. So no problems with interrupting this. When Peter roped me into this, he had already assigned the normal machine learning multivariate pattern analysis talk to Martin, which is both a blessing and a curse. The blessing is there's lots of things that I won't have to cover, but that is if you are actually at Martin's talk. Were you? Nope. All right. That complicated things, but it's useful to know. The second thing I wanted to say is, while ostensibly this talk is about encoding and decoding models, it really is an excuse for me to rant about my favorite quote. And it's not just my favorite quote, because it's a really good quote, and I've been at a workshop where every single person speaking, 6 people, all had this quote in their talks without coordinating. So really, this is a useful piece of advice. The second thing I had to say was this, but I already went through that. So the reason I had that quote there was the following, is in this talk, all the things I'm going to cover are approximations to something that the brain is doing. And the talk really is about how do you torture a model, or how do you set up a model so that you can draw conclusions about the things you're really interested in. And this is valid for GLMs, this is valid for vanilla decoders, this is valid for encoding and decoding models. The difference is that in encoding and decoding models, you actually have a chance of getting closer to the representations that you care about directly rather than by deduction. But rather than pontificate without slides, let me be more specific. So if you are running a GLM, which most of you will have done at some point, you are feeding the subjects a visual stimulus. That's what you pass to the GLM. The GLM does a prediction of every voxel in the fMRI you collected during the experiment. You get some p-value map. I'm glossing over having multiple subjects, but you get some active locations at the end. And that's really the point, is you go from something that's being shown to some notion of location. It's mass univariate because you fit every voxel separately. Testing, you take into account the fact that voxels that are nearby are doing similar things. There's other refinements like having nuisance regressors like motion correction parameters, or having some kind of graded response in the stimuli. But in practice, it really boils down to a good GLM explains the behavior of voxels somewhere. Clear? If you instead are using a machine learning classifier of some sort, you are training a decoder to look at training data where you have some label, which is a stimulus the subject was seeing, say, and the decoder looks at lots of voxels and predicts what that label should have been. And it does this, hopefully, when faced with images it does not know the label of, and it can actually get the label out. So Martin's talk was all about this. If you're interested in this stuff, I heartily recommend his talk. It's really, really well done. But by decoder, I'm actually being pretty loose because it could be prediction of some measurement about, say, the subject's intensity of subject's emotional reaction to a stimulus, or anything else you might want to predict from the images. It is multivariate because you look at many voxels at once, and a good decoder is one that's accurate at predicting the right stimulus or picking the right one out of a lineup. All right, so the reason I'm going over this so quickly is I really want to get to this slide, where the thing that really matters here is, yes, you're showing subjects some stimulus, stuff happens in here, and you collect a functional MRI. And both GLMs and decoders are nothing more than tools to ask one question of the data. So in the GLM is there a brain location that responds to the stimulus? In the decoder is there information about the stimulus in the pattern of activation? And you can view it as going in this direction or going in this direction. And that's really all there is to them. And so the reason I'm beating on these are tools is that people have this unfortunate tendency to start treating these things as some kind of analysis path on where there's only one true way. That's not right. These things are tools for asking questions, and they're either the questions you want or there are other questions and you have to figure out a way of going to the ones you really care about. The reason I also dislike this is that if you think about these as tools for answering these questions, that's where you get these caricatures from, of GLMs as modern phrenology, or decoders as some form of parlor trick for doing mind reading, which I think these caricatures are sad, but often they are actually quite accurate when people treat this without being critical in their thinking. So the questions we care about, as you well know, is we want to know, what does the brain area do? Is the brain area used in a particular task? If they're doing a certain task, what are they representing in their mind? Does it differ from subject to subject depending on some characteristic? Are those representations invariant to things like position of an object you're seeing at? Is there a place that's sensitive to position? Is there a place where you are only sensitive to the identity of the object? These are the things that we really care about in terms of doing cognitive neuroscience or any clinical application. And so how do we go about answering these questions? For a GLM, it's fairly straightforward because people have been doing this for a long time and they've been critiqued enough with the phrenology thing that they've started thinking more about what they're doing. So you do this by being careful with how you choose control conditions so that you can find a control that differs from what you want as little as possible. So if you're studying sentences, you might have words, you might have scrambled words, and then as multiple conditions to control against the one you really care about. And the other thing is actually, assuming you have some statistically valid result for there's activation in this location, is using some form of meta-analysis to perform what's called reverse inference. And this paper by Russ Poldrack is fantastic at explaining this. If you haven't read it and you do this kind of work, you really, really should read it. Because the point he makes is that knowing the answer to this does not give you the answer to this. So how likely is it that you're doing a certain task given that you see activation here? How crucial is this area where there's activation for the task you're doing? The way you do that is by going to some form of activation database where people stored activation locations for thousands of studies or statistical maps, and those can tell you how many different tasks actually use this location that you just found. And you do better by being careful with your controls, narrowing where your activation is, and also narrowing down the sort of tasks that you care about. And there's different resources with different trade-offs. More than happy to talk about this if you want at some point, but they're all worth checking out. The reason I'm not going to spend a lot of time on this is that otherwise I would never get to encoding models, and there's still decoders along the way. But any questions about this? Does this make sense? Okay. If you're using decoders, you have the same problem. You're not directly answering any of the questions you care about by using a decoder. You are answering the question we had before. So how do people do it? They do it by using one of these three approaches, and rather than read them out when they're in front of you, I'm just going to give you a nice illustration of each one of them. So the first one is, as Martin had on his talk, is you restrict which voxels you actually feed to the decoder. So you could use the whole brain, and that gives you one result, says that yes, somewhere in the whole brain there's something that allows you to predict what the subject is doing, or how they feel about the stimulus, or something of that sort. And if you don't care about localization, this might be okay. Or you might just restrict to regions of interest where you a priori have some reason to think the information will be there. And this way you can actually compare different regions. Or in the extreme, and then you get one accuracy per region. Or you can use a searchlight and get an accuracy map covering the entire brain. There are trade-offs in doing any of these things. But this is exactly the sort of thing that the machine learning team deals with. So if you want to do searchlights, come talk to us. We can discuss all of this with you. But this, for now, is just an illustration. The other way you can get answer questions is by selecting your input voxels by how they behave. And in this case, you could find voxels that you just don't want to use the entire brain. You just use voxels that are active during the task, no matter what they're doing. They're just doing something. You have two conditions that you want to distinguish, but as long as it's doing something versus baseline, you pick the voxel. Another way is to say you want voxels that are not noisy. So they might be voxels that respond consistently across different conditions. They might not be doing anything at all, but that means they're consistent in what they do. Or you could go further and actually say, if I want to distinguish A from B from C, then you might pick voxels that are selective for each of these conditions in isolation. Of course, if you have many conditions, there's probably no voxel, there's no grandmother's, well, we're not sure about grandmother cell, but certainly there's no grandmother voxel. So you are only going to have, you might have just situations where voxels respond differently to some conditions than they respond to others. I mean, these are all things that you can mathematically define and identify. There are some dangers of circularity, but that's also a separate topic. I think by now there's enough public awareness of that people know what the dangers are. Otherwise, if you're wondering, is there circularity in my analysis, please do come talk to the machine learning team. And finally, I'm getting to decoders, to encoding models in a few seconds. The last thing that I wanted to show you was a more creative use of classifiers, which is treating them as some kind of virtual sensor. So the idea is to say, this is going to be from an experiment that looked at free recall, but before they do that, they make people are studying items that are either faces, locations, or objects. And they do this in blocks in the scanner. So in every block, you know what the person is thinking of, so you can train a decoder that says, are they thinking about faces versus everything else, or locations, or objects? Does this make sense? So this is easy to train, it's block design, the signal is really solid. But then, once they have these little classifiers, they use them as a kind of sensor of what the subject is doing at another time. So this is another part of the experiment where subjects are doing free recall in the scanner. This is time. And what's plotted here in the little dots is just when they started recalling items of a given category. Now, the cool thing here is that these things kind of cluster, but the hypothesis they wanted to test was, is it really the case that you get some reinstatement of the pattern seen during study at the point where you're having a recall moment? And you collect fMRI data throughout this period, you apply each of the classifiers to the data, and at every point you are plotting in red or blue or green how certain each of these classifiers is of whether it's seeing a pattern that matches what it knows about. And the cool thing is that this was sort of shifted to account for the hemodynamic response, that at the time you're having a burst of recall in one category, the confidence of that classifier also goes up. So does this make sense? I mean, I'm treating the talk also as an opportunity to show you some of the more creative designs I've seen, because just to give you a sense of what the tools can be used to do. But this is just with vanilla classifiers. And once you do this, you can also ask yourself, in space, where do these classifiers pull the information they use from? And it's different locations for different classifiers. So this is all possible using these methods. So it's not like you can't answer questions using GLMs or classifiers. Of course you can. But the issue is the inference is rather indirect. And so in GLM is you are kind of filling with the contrast to see what happens in the subject. In the decoders, you are using feature choices, and then you dissect the decoders to understand what they're doing. So you could pick different caricatures, be it the frog legs as you're just poking and making something happen, or dissection of some sort. But the thing is, it doesn't really have to be. And this is what the rest of the talk is about. So the rest of the talk is about models where, knowing that this is the situation, you're willing to say something about what the representation is of the task, of the stimulus, of intermediate things that the subject needs to derive on their head, and test that. And what could this be? I mean, it could be something you know because we've dissected enough brains by now that we know something is there. It could be constrained by behavior, by animal experiments, and there's lots of clever behavioral designs that tell you that the brain represents one thing or another thing. It could be a mathematical computational model that says, given the stimulus, you will build this data structure in your mind if you are going towards cognitive psychology. It could be a hypothesis. Or it could be something that's learned elsewhere, and I'll tell you quite a lot about this further down the line. But there's many ways in which you could get some hypotheses about your internal representations. And then the question is, how do you use these and imaging data to test those hypotheses? The earliest version of this was something that Nico Kriegskorte did with looking at many stimuli in several different categories that have some taxonomic relationship. These don't matter too much. The point is, for any of these stimuli, you get images, and you can look at their correlation of those activation patterns, and that gives you some similarity matrix that has a structure of some sort. I mean, this is pretty old by now, but the reason this is kind of neat is that it's looking at the signal, and then you can use this idea to test hypotheses such as, if you look at patterns of activation in different parts of the brain, does the similarity structure across the exact same stimuli change? And that similarity structure changes from location to location, or from chunk of the ventral visual stream to other chunk. And so contrasting different locations or different stimuli allows you to do inference on what is actually represented. So it's possible to do this with nothing more than having patterns of activation. However, once the things you're trying to train are a bit more complicated, what you can do follows in sort of two general cases. One is an encoding model, which is where the question that's asking is, does my representation predict brain activation? So what we're doing is we go from stimulus to how it's represented and some mapping from the representation to the image, or the reverse, where you try to infer from the imaging data via a mapping what the representation is, and maybe even reconstruct what the stimulus is. Does this make sense? Okay. And again, rather than pontificate over one diagram, I would rather show you some cool case studies that I think make the point better. So the first one is for an encoding model. Again, a paper from most of these papers will actually be from 2008, which was when there was a burst of interest in this type of model. In this case, the authors were looking at 60 different words in 12 categories, and in every trial, subjects would see a line drawing and the name of whatever that object is. And the model they built, so we're going from stimulus to representation, is a vector of semantic features that represents aspects of the meaning of this particular concept. I'll tell you what these are in a minute. Then the mapping is just a prediction of every voxel in the pattern of activation for table based on these semantic features as a linear combination of them. So far so good, but I haven't really told you where these things come from. And this is also to give you a sense of how disparate a source you could have for the representations. In this case, given that the goal was to represent different aspects of the meaning of a word, they decided that some of the important aspects might be sensory or motor or physical in some way or other topics. And they decided that for every aspect they want to consider, they would use a verb as a proxy. And the way they figured out how to represent each noun is by seeing how often does it co-occur in a sentence with each of these 25 verbs in a very large text corpus. Does this make sense? And so that gives you numbers. So airplane co-occurs pretty often with ride and less often with a number of other things. And so each of these numbers becomes that semantic feature in that vector I was showing you earlier. But then how do you go from this to having a mapping? For that, let me just give you, this is a caricature of a pattern of activation where different little spots with different shapes are just things which are present in that pattern of activation. I never figured out why PowerPoint does this to me. It's profoundly embarrassing and it happens every single time. And short of killing all of them, I don't really know what to do. You can tell me afterwards if you know the answer. The main intuition is this, if two things like table and chair are related, their semantic feature vectors will have many things in common. This you can test experimentally by looking at semantic feature vectors. You also hope that table and chair, by virtue of being furniture, also have something in common in their patterns of activation. So in this case, this red circle and the green triangle. And hammer, maybe by virtue of being an object, has something in common with, actually no, not being an object, something else. So actually by virtue of being all of them being objects, they all shared this red thing, but they don't share other things. The idea being that there's something in corresponding between sharing semantic features and sharing brain activation, which is a reasonable hypothesis. So then, if that's the case, and I'm just going to unfold each of these into the individual components, you can look at these and actually tease apart each of the patterns of activation into component images, basis images, associated with each of the semantic features. So if it's present, this is present here. If this is present, it's present in all of them. Does this make sense? I'm not having any math here, but basically you can take my word that this is just a series of linear regressions. So you can do something like this, and given a set of training images and the labels and the semantic features, you can learn these basis images. So in practice, it might look something like this. Here are 3 verbs, here are the corresponding images, and the basis images just basically capture how much does each semantic feature affect activation in one part of the brain. And then you can predict each, you can look at a certain, if you want to predict what the activation would look like for a new word, you look at its semantic features derived from text, you look at the corresponding basis images, you add them up, and you end up with a prediction of what the pattern of activation for celery would be like. Does this make sense? Good, because it will be like this for every single study till the end. People are not super creative about this. There are some deviations into nonlinearity, but most of the time this is all that people do. So, okay, you have a model that comprises a representation and a mapping. How do you know the representation is actually doing anything? In here, they kind of eyeballed, actually in the previous slide, they kind of eyeballed it and say, well, eat kind of uses this bit, and push will actually use postcentral gyrus, so you could imagine there's some sort of motor program being activated. You could hand wave this way. Or you could say, what happens if I actually throw my model at words it has not seen before and used in the training set? So in this case, they use 58 of the 60 words to learn the basis images, and they use those basis images together with semantic features for 2 left out words, in this case celery and airplane, and predict what those images should look like based only on information from the semantic features derived from text. And then they compare, does the predicted image for celery look more like the actual image for celery or the actual image for the airplane? And if it looks more like the correct one, you count this as a success. Otherwise, you failed. Does this make sense? So the key idea I want you to take here is less about specific accuracies or anything like that, is the idea that what you do with these models is you make a model, and then the model should allow you to go all the way from one end, stimulus to activation, or activation to stimulus or representation, on new items it has never seen before. And that's what allows you to validate the model and say, yes, my representation learned something that's actually in the brain. The other thing I want you to take from this study is that you can learn the basis images, and faced with a new brain activation pattern, you can say, I'm going to decompose this pattern in terms of what semantic features are present for the basis images to work out. So something like this. There's a little yellow thing, this basis image is there. There's a red thing, this basis image is there. Other things, they're not. So any encoding model, or most of the ones you'll encounter in literature, can be inverted without any problem and be used in the reverse direction. And this is also sort of an important thing that articles don't always talk about. Any questions about this? Yeah.
This is all like within person, right? So you would train on one person. The accuracy is averaged across within person or something.
Exactly. There are models that work across people, but those, in all of those, they tend to find basis images or things like that correspond to things like food, shelter, tools, and you think these things are really important. Well, they are important, but but you also think maybe these are the only things that give rise to large enough blobs that if you overlay everyone's brains, the activation is more or less in the same place. There are other ways of working with multiple people to find what's common, even if the locations, so they might be representing the same things, but the locations are slightly different. And if you have something like that, feel free to talk to us, because we have code for doing that. Any other questions? Okay. So the second case study, really, really famous, is basically one where the authors wanted to test a model of what was happening in the early stages of processing of visual stimuli. Now, this is from 2008, which is right at the beginning of the deep learning revolution. So the models that they use are actually now kind of quaint and cute because they're sort of human specified. But at the time, they were pretty cutting edge. We'll talk about the ones derived with deep networks later. The design was pretty simple. People were seeing stimulus images, grayscale, for a few seconds and then off. And they had these images, these images for training, these images for testing. And the model that they used was the following. For the representation, they would just take a series of Gabor filters at different scales and apply them to the visual, to the image. And so the representation really is the output of all these different filters. And then they predict what every voxel is doing in response to the stimulus by combining all the filter outputs that you have. So again, this looks exactly like the previous thing. Before you had semantic features combined into what a voxel does. Now you have filter outputs, linear combination into what a voxel does. And the reason I like the previous study is that it's easier to explain what's happening, but the mechanics are exactly the same. Then, just as before, they want to evaluate this, so they take their test images, they derive the representation by passing them through the pyramid of Gabor filters, they predict the activation, and they classify every one of them by how similar it is to the actual activations. They get pretty high accuracy, which is a sort of a testament to how nice their data is, as much as how nice their model is. But it's a really impressive study. The reason I'm not spending too much time on this is I already beat you over the head with all the details of how to test a model. So I would rather go to a decoding part done on exactly the same data. So the model there starts by being exactly the same, and so is the mapping, but they add the semantic category labels for each one of the stimulus images. So now your representation is a mix of visual information and semantic information, just which category the thing is actually in. And the way they try to, so their ultimate goal is to say, take this stimulus image and find me on a database an image that looks like it and has broadly the same things in. They call this a reconstruction. You could argue how much of a reconstruction it actually is, but it's still pretty impressive that you find something like that, you aim for something like that. And just as I showed you before, as in a new test image, you could get the semantic features based on the pattern of activation. They can invert the model that they had, the combination of Gabor filters and semantic information. It's more complicated than just doing the inversion I was showing you earlier, but there's no point in dissecting the model here. The idea is the same. Then you apply it to the activation data for a test stimulus, and you get some kind of probability for each image on a really large database, millions of candidate images, for how likely is it that this image could have been the stimulus that the subject saw, given the activation that was collected while they were seeing it. And then they evaluate this by seeing if the semantic category of what you found actually matched that of the stimulus. What's kind of neat here is that if you just use the visual information, you'd get things that arguably had some shared visual statistics. Not entirely sure I buy it, but this is the figure they have on the paper. But if you use visual plus semantic information, you actually get something that's thematically closer and also visually closer. So their claim here is that, yes, in the areas they're looking at is they're using the visual part in one part of occipital cortex, and the semantic part is really using voxels that are further along the ventral visual stream. But if you combine these two types of information, you can perform some sort of approximate reconstruction. And given that I've just been hand-waving about the ventral stream, the third thing I wanted to show you is on the same data. Yeah?
Interesting. So with that study, you mentioned that kind of it was pretty close to cutting edge for the time, but there's a lot better stuff now. I'm trying to think, has anyone taken that data or taken a similar thing and essentially done better with the same, with more modern methods?
Yes. Good question. Dan is not an audience plant. Yes, and I have to thank Patrick for pointing me to this last night, because I was thinking someone must have done it. I was asking myself the same thing, and Patrick just had the study like that. Yeah, these guys were reanalyzing this data set, and instead of using the Gabor filter pyramid, this is after the deep learning renaissance and the development of large image databases and imaging classification competitions. So now we can go from the stimulus through many layers of a convolutional neural network that are essentially applying filters. But now the filters are not specified by humans. The filters are learned from data to produce a series of layers that correspond broadly to the things that you had with Gabor filters. But they're all learned from data to do classification of natural images into many different categories. And then this can be coupled to a mapping where, again, every voxel is a linear combination of network outputs at different layers. The original network, if you're curious, was trained to, given an image, learn to filter it so that at the end you can classify which of 1,000 different categories it could have come from. So that's what the network is trained to do. But it learns pretty useful representations at various levels of the input image, and those can be used to learn a forward model. So the most interesting thing in their paper is not that they can do accuracy X or Y, it's really the way that they try to validate representation. And they do this. They can look at each voxel and see in the test stimuli, let's try to predict it using each layer of the network in isolation. And then you can see, for this particular voxel, over all the new stimuli the model hasn't seen before, how well is it predicted by layer 1, layer 2, layer 3. And so the layer that best matches a voxel is color-coded here. So we go from V1 all the way up the stream. So the neat thing is that the voxels that are further along are predicted better by the later layers of the network. And so going from V1 to V2 to V4 and so on, you start to see more and more voxels that are better predicted by later layers in the network, which is the layers where you have some degree of invariance to rotation, to the object position, to some specific details. So it's kind of really neat that you can go and find this in brains once a network has already found it for you. Any questions about this? Yeah?
So if I understand correctly, those bluer areas represent like low-level visual features of the stimulus.
Yeah.
Versus something that's more categorical.
Yes. Yes, much better put than I did it. Thank you.
Thank you.
Any other questions? If this wasn't clear. Yeah. I think it's a really, really fantastic figure, and they replicate it across 2 different people. The last study I wanted to show you was something sort of going back also, just like this one went back to the earlier data set, going back to the data set from 2008 with all these different items in a categorical structure. So there they had found that in human IT you had this similarity pattern across the different types of stimuli. What this study did was to see how much does this similarity pattern that we see in human IT match the similarity patterns that you would see if you passed these stimulus images through a network like the one I just showed to you and looked at the patterns of activation in different layers of the network and looked at the similarity between them. So they derived those, and it's kind of neat that as you go up the network, you start seeing some of the macro structure that's in here emerge. Of course, one of the reasons this study is interesting is that it's not like you get the exact thing you represent in IT out of a network, because that would be too much. I mean, yes, if you got it, fantastic, but it's not expected. You get some of the structure. Some of the structure will be in some layers, some in others. You can kind of squint. Or you can do what they did, which is to say, well, the model that I was showing you in the previous study uses a network trained exclusively on natural images from an image analysis challenge. But you don't have to take that network. You can tweak it during training so that layers match more closely what human IT does. So you can get parts of the network where the similarity structure actually maps what you have in humans, and that network is still capable of doing most of the tasks that the original one did. And this is maybe the other thing that I wanted to stress, is you're not even though many of the interesting representations we can use come from other domains, text corpora or image databases or whatnot, you're not a prisoner of those. You can, in your training data, make things match what you want. You can sway the network in a certain direction, and later when you test on new stimuli, the network will have the properties you want. So that's also open to you. Any questions about this? Okay. So just to give you a sense, I could have highlighted many different papers, and this has been, the reason I'm putting them all here is not to fill the slide, but so that you can kind of see that there are nice models for a lot of different types of stimuli. And of course, this is pretty coarse, because you could still say, well, if you have a story, do you just have the words? Do you have indications of which characters are appearing? Do you have indications of the emotion that's present in a scene? Do you have an indication about whether the scene is set inside or outside? All of these things can now actually be automatically generated for a lot of text, movies, etc. Just exposed to Talia Arconi's NeuroScout, which is really nice and can actually do this for a lot of stimuli. But the other thing that's kind of simpler is that it's often easier to do encoding than to do decoding, but there's quite a few that do decoding and even try to reconstruct the stimuli. I have less things here in representational similarity because this is where the papers vary a bit more in how they go after the similarity, but these are very good starting points, and anything that cites them is probably, if these are of interest, you can find the other ones. So to conclude, I want to just give you a very quick summary of what to take from this lecture. The first thing is there's lots in common between these models, and the representation that they use is usually pretty complex. So you are not forced to looking at predicting one number or one label from a set you already know. You can find something that's probably arbitrarily complex at this stage, and any type of naturalistic stimulus, there will be a database somewhere we can use. And the same representation can be used in either direction, encoding or decoding. Whether you do so kind of depends on what you want to do. And that's the second part, is you really should think about this paradigm as you have some training data and you only trust generalization to stimuli you've never seen before. So you use some data to learn mappings and others to evaluate. And when you're evaluating, you could predict imaging data, which is okay, and you can try to infer some representation, and these can be analyzed by comparing to actual imaging data or to other similarity between representations that you know from some other source. In the limit, you could reconstruct the stimulus the subject saw up to a point. And for this, having some prior information helps. As I was showing you in that database, if you know the type of image it is or semantic content, it's much easier to narrow down the choice. But whether you want to do this is also a bit of a gimmick. So there might be the question I want you to take is if what I want to do is retrieve some aspect of the stimulus from the imaging data, that can be done. In terms of the differences between the models, is the encoding models are really good for trying to say, maybe you have different competing models and you want to see where are there voxels or locations that this model can actually predict. Jack Gallant does fantastically colorful maps in his papers, all based on whether a voxel can be predicted or not. And this is useful for testing competing theories about what is represented where, and as long as those theories can be turned into representation, you can try this approach. And then, if you need a number, because the reviewers are going to ask you for a number, you can classify predictive activation by how similar it is with the true activation. If you're doing decoding, that's the right thing to do if you need to extract a representation from activation for novel stimuli. Maybe you want to say, I think that this is represented, but not that, and you carefully pick your stimuli so that you contrast the things that you want. This is when you'd want to do something like this. And in the limit, you can reconstruct a stimulus too, or an approximation, if you really want to do that. If you want media attention, yeah, do try to do that. And then I also want you to think that you can use ideas from representation similarity analysis, and this can be done either in encoding or decoding model, because you can compare activation in different locations, you can compare predicted activation, you can compare the representations and match this to some information about similarity of stimuli that you got from some other source. With that, I think I'm at the end. I just wanted to advertise for our team, because we can definitely help you with turning stimuli into representations. Sometimes it can even be done automatically, other times it can't, but we should at least alleviate the effort put on your ERTAs. We can help you derive representations from behavior and from other sources, and help you devise some kind of encoding, decoding model strategy if that's what you need. Or we can help you using all the methods I described in the first section of the talk. So that was an intro, but also a little bit of an advertisement. The thing we can't help you do, I actually had, we can help you train networks, but then I thought, no, we can't. That's just a little bit too hard, but we can give you some tips so that you don't get stuck. And so either email me or drop by, because I'm usually in the office. With that, thank you very much, and tell me what questions you have. There have to be questions.
So you mentioned at the beginning of the talk that some of the things we're interested in the field are seeing how neural responses in certain brain regions are invariants of other processes, or using some of these tools to identify clinical outcomes. As a lot of these models seem to be kind of specific to the subject that it's trained on, do you have any thoughts on how to best use these sorts of tools in clinical research?
I almost didn't put the clinical question.
Can you summarize the question for purposes of the recording?
Oh, right. Let me see if I got it right. So basically you're concerned whether, because these models are all specific to a single subject, whether they would have any practical validity in a clinical setting.
I was curious as to that.
Yes. No, it's a legitimate question. I almost didn't put anything that said clinical in the talk, because I thought every example I have is really cognitive neuroscience. And for most of the clinical papers I'm thinking of, they're really not looking at activation. They're looking at connectivity of some sort. They're looking at features derived from structural images, because those are much more easy to equalize across subjects, or to understand how do they vary due to subject characteristics or due to the disease at hand. But to pick on the question that you also had earlier is, if any of what I was showing had clinical validity, there are ways of saying there's a representation. We know it changes in this way for different stimuli. We hypothesize that everyone has the same changes. And we can say, okay, the activation in response, let's say that you're looking at positive versus negative stimuli. And your interest is, say, maybe in depression. And you want some kind of screening. So, I hope this doesn't turn out to be a stupid example in the end. But you could say, yes, you have a sequence of stimuli. You expect that everyone would react to them in the same way. Then you can say, there's maybe sort of two different variables under here, positive and negative. And if it's positive, you're going to see activation broadly in the same area, but in different voxels in different people. it's possible to learn a model that identifies for these shared variables that describe the stimuli in each subject which locations carry information about them. So if you use something like shared response modeling or generalized CCA or even hyperalignment, they will all give you a variant of this. And then you can get clever with, you find things in common between patients and controls, or you find things that are only in the patient group or only on the control group. So if I had to use activation for clinical purposes, I would say that's what I would do. It's actually what some people we know are trying to do.
Thank you.
You're welcome. Adam? Any other questions? Okay, thank you very much for listening to me.